Okay, this is chapter 14, Mendelian genetics. And we're going to talk now about more examples of using Mendelian genetics in Punnett squares. Okay, let me share my screen. Okay, so we left off last time at learning on Punnett squares. Remember that the gametes, which only have one copy of every allele, go on the axes, right? And then they come together in fertilization to create your diploid offspring, right? And these are not necessarily saying all of these offspring are going to be made. These are all of the possibilities and the predicted genotypes. And that genotype, again, can predict the phenotype. So let me organize my screen a little bit. Just a minute. Okay. All right, so here we are now looking at what is called Mendel's law of segregation. All right, so we know that there are two alleles for every observable trait, right? And why are there two alleles? Because we are, you got it, diploid organisms, right? Those two diploid organisms occur in homologous chromosomes, right? Homologous pairs that get separated during meiosis 1, and they get packaged into our gametes. Whoops. Remember, the gametes are haploid. They only have one copy of every gene, right? So Mendel's law of segregation really describes how you go from diploid organisms to haploid gametes, right? It really kind of describes what happens in meiosis. So once scientists really understood meiosis and gametogenesis, which is the production of gametes, it really supported this law that you have pairs that get separated, right? Punnett squares are then used to predict the offspring. Okay, so let's do an example here. So we're looking at a different example. We are looking at yellow peas and green peas. We're going to make the assumption, or I'm going to tell you here, that in the pea generation, these are both true breeding. So remember, that means that they are homozygous, two copies of the same allele, right? So we have a yellow, which is going to be big Y for the genotype, and green, which is little y. And we're going to cross those together. So each parent is homozygous, meaning they can only produce one kind of gamete, right? So the gametes are going to be, you can only make big Ys for the yellow, and the green plant can only make little Ys. So that means in the F1 generation, the offspring are all going to be heterozygous. These are the hybrids. You have one of both alleles, right? And those offspring can make two different kinds of gametes. You can make a big Y or a little y. And again, this has to do with the law of segregation. You go from having two copies of every chromosome to one copy when you make your gametes, right? You go from diploid to haploid. So your job is to do the F2 Punnett square. We're going to talk about what the phenotypic ratio is, what the genotypic ratio is, what is the chance of seeing a dominant phenotype, what is the chance of seeing a recessive phenotype, and what is the chance of each genotype. All right, so let's go ahead and do that together. So I'm just going to draw this out. So we had big Y crossed to, little y. And again, if we do that Punnett square, I always make my little letters cursive so I can tell the difference. All right. So if we do this, what you'll see is that we're going to get 100% heterozygous, big Y, little y. And those are going to be also 100% yellow because we have that big Y there, right? So these were yellow and these were green. Okay. So now what the question is asking you about is what happens when you cross two heterozygotes together, right? So what happens when you do that, when you make an F1 cross? So again, the first thing you need to figure out, what are the gametes that this one can produce? It can produce either a Y, big Y, or a little y. Same here, you can either make a big or a little. So we're going to do the Punnett square. And now we can go back and answer that question. So I'm going to kind of toggle between. So what is the phenotypic ratio, right? So the first question, phenotypic ratio, means how many yellow to green do we have? And we're going to have a 1, 2, 3 yellow to 1 green. So that's going to be the phenotypic ratio. Next, what is the genotypic ratio? So here, remember, we're going to have the genotypic ratio being big, big, big Y, which we have 1, big Y, which we have 2, little y, which is 1. So we have that as the genotypic ratio. Third question, what is the chance of seeing the dominant phenotype? So which is the dominant phenotype? The dominant phenotype is what you see after you cross those true breedings, right? It's the one that is the dominant. So in this case, that is going to be yellow. And what is the chance of getting yellow here? It is a 75% chance of getting yellow when you do that F1 cross. What is the chance of seeing a recessive phenotype, right? So the recessive phenotype is the one that is hidden in that first cross, right? And then reappears when you cross the two heterozygotes. So in this case, it is going to be green, and it is a 25% chance. And then the last question, what is the chance for each genotype? Okay, remember genotypes refers to the letters. So the chance that you're going to see a homozygous dominant, meaning big, would be 25%. What is the chance that you'd be heterozygous? Would be 50%, right? Because you have 2. And what is the chance of being little y would be 25%. Okay. All right, what we've been doing is referred to as complete dominance. In other words, this is traditional Mendelian genetics or Mendelian inheritance, right? All of these, when you cross two heterozygotes, you will get a 3-to-1 ratio, right? So this is when you cross. When two hets are crossed, you will always get a 3-to-1 ratio. All right. And this occurs in seed shape, pod shape, flower color, seed color, pod color, stem length. Turns out that pea plants are very, very predictable because almost all of their traits are determined by complete dominance or very predictable Mendelian genetics. Again, showing what an awesome model organism that pea plants were for studying this. Okay, so there's a few ratios that you're going to have to know. One of them is this 3-to-1 ratio. When two heterozygotes are crossed, you get a 3-to-1 ratio when you're looking at a monohybrid trait or when you're looking at a single trait. All right, so I've been throwing out these words dominant and recessive, so I want to actually define them for you. So the dominant trait is the one that is expressed in that F1 generation when you have an F1 generation, and I'm going to put after crossing to true breeding organisms, right? We represent that as the capital letter, right? The recessive trait is hidden after crossing to true breeding, right? And that is represented, sorry, it's for some reason it's down here, by the lowercase letter. So if we just look back at our example, we had, just as a reminder, I'm gonna do that over here. Oops, sorry. I'm gonna do that over here. So remember, we had big Y crossed to little y, right? That gave you our hybrids, which were all yellow, right? So that means that yellow is the dominant trait and green was the recessive trait. In general, students are always confused on like, what letter should I use? And in general, the letter represents the dominant phenotype. So big Y stands for yellow, right? And that's the dominant trait. All right. So we know, for example, so if you look at a plant or in this case, a pea plant, color, flower, we know the phenotypes of them just by looking at them. We know whether it's purple or white or green or yellow, right? But we don't necessarily know the genotype always, right? For the white plants, we always know the genotype. Because it's the recessive one here, we know that the only way you can get a white plant is by being little p, all right? However, the purple, if we just pick up a random pea plant that's purple, we don't know what its genotype is. We know its phenotype is purple, but we don't know its genotype. It could be either big P, or big P, little p. It could be homozygous dominant, or it could be heterozygous. We don't know. How would we figure that out, right? We would do what's called a test cross. And a test cross is taking that plant that we don't know its genotype and crossing it to something that we do know the genotype, such as a white flower, right? So let's go ahead and do those two crosses. So what we're trying to do here is determine which genotype does a purple flower have, right? PP, big P, or big P, little p. That's our question. So what we can do is cross that, let me hold on, wait for it, my purple flower. We're gonna cross that purple flower to something that we know the genotype. And for this, we're gonna cross it to a white flower, right? So look at the two possibilities. We can have, so what we know is that we have our genotype for the white flower, and we know that it's big P something, right? So let's just try it out. Let's just see what we get. So if it was homozygous dominant, big P, then our outcome would be 100% purple flowers, all heterozygous. If that purple plant was a heterozygote, we would get a Punnett square that was fundamentally different. Notice now we would get 50% purple flowers. Whoops. Oh, come on. And 50% white flowers. So by doing a test cross, taking that purple flower and mating it to a white flower, we can tell what that original parent was, depending on the outcome of that cross, right? That's a test cross. Helps you determine if a dominant phenotype is homozygous dominant or whether it's a heterozygous, based on what you get out. All right, so that was all about monohybrid crosses, looking at a single trait. Now we're gonna delve in to dihybrid crosses, two genes. or 2 traits involved. And for some reason, students always, their minds explode when we go to 2 traits, but it shouldn't. Just remember your fundamentals, all right? So first off, let's look at what the gametes would be for the following genotypes. And students always are like, I don't know. Take it slow, go gene by gene, right? So for our, yes, I am, Bex. My goodness. He's like a bull in a china shop. All right, so here we are looking at smooth yellow, right? So that is the phenotype, is smooth yellow. The genotype is big S, little, or sorry, big S, big Y, big Y. What are the gametes that can be produced? So remember, each pair is going to segregate, the law of segregation. So the gametes are going to be one S, because those S's are going to split up, and one Y. That's the only gamete that can be produced by that organism, right? If we look at this other true breeding plant, which is wrinkled green, little s, little y, the only thing that you can make is little s, little y. That's the gametes, one copy of every trait, right? So you're splitting up your homologous chromosomes, get separated. All right. For the third one, we have little s, big Y. So the little s's get separated, so you can only give little s. The big Y's get separated, and you can only give big Y. All right, what about smooth green? Right, you're seeing a pattern here. So here's the one where people get really confused, all right? Take it slow. So you can either give a big S, right? Or you can give a little s. You can either give a big Y or a little y. So you can have big S, big Y. You can have big S, little y. You can have little s, big Y. You can have little s, little y, right? So you can remember one copy of every chromosome, of every gene. Let me give you one more. What if the genotype was this? What are the gametes that can be produced? You can give a big S and a big Y, or you can give a little s and a big Y, right? You have to be able to understand the gametes before you can do dihybrid crosses, right? The key here is that the gametes must contain one copy of each gene. That is the main point here, all right? So what we have here is that we are going to be looking at an example where we are taking true breeding yellow and true breeding wrinkled green, sorry, smooth yellow, wrinkled green. Those are both true breeding, meaning they're homozygous for both genes. And so if we look at their gametes, this smooth yellow can only make big S, big Y. The wrinkled green can only make little s, little y, right? When they come together, you are going to then re-put in, you're going to fertilize those. So you're going to make those the egg and the sperm are going to come together during fertilization and restore the diploid state. So we're going to now have big S coming back together with little s, big Y coming back together with little y. That's going to be our outcome, right? Our F1 generation, right? You can get that by doing this cross. You could also get that by doing this other cross, right? And you should look at the genotypes to convince yourself of that, right? However, now we are going to cross those F1 generations together, right? So let's look at what that looks like. So we are going to be crossing two, I'm going to say two, I don't like doubly, two, and I'm trying, sorry, two plants that are heterozygous for both genes. Whoops. Okay, we're going to cross them to two plants, heterozygous for both genes crossed. All right, so what we had was big Y, big S, little s, crossed to big Y, big S, little s. Okay, so here's our cross, right? These are the genotypes of the parents. First thing we have to do always is determine the gametes. So what are the gametes that can be made? You can make big Y, big S, big Y, little s, little y, big S, and little y, little s. And you're going to see the same thing for the others. And just for ease of writing these out, both can make those four gametes. So what is our Punnett square going to look like? You guessed it, a 4x4, right? So we're going to see a 4x4 Punnett square. Are you going to have to be able to do this? Yep, sure are. So you might as well get practicing, and it just takes practice, all right? What you'll see then is when we put these together, we are going to see that we're going to restore the diploid state each and every time. So we're going to put the big Y's together. And some students shorthand this and they never fully understand it. So it's really important to write all of this out. Sorry, that's a little less. So that you fully understand your Punnett squares. All right. So I'm just writing all of this out. You should also, in your notes, be writing out the full Punnett square for a dihybrid cross. We are looking here at two genes. Notice how I always make my small, my recessive trait, my small letter cursive. It really helps to tell the difference. If I can't tell the difference between your uppercase and your lowercase, I can't mark it correct on a test. So really be careful with your handwriting on these. SS. Okay, so notice we have 16 possible offspring. All right, 16 different possible offspring with a variety of different phenotypes. Okay, so here's our dihybrid cross that we just did. Notice again that we have our gametes, which we accounted for, right? And we have all of the possible offspring, right? Now you can calculate genotype and phenotypic ratios, right? So how many, for example, big S, big Y are there, right? And it looks like only one, right? And you can go through and predict that. So how many, for example, little s, big Y. So you go through little s, big Y. Okay, so there's two of 16, right? All right. The kicker is that you also have to be able to determine the phenotype that comes out of this. All right. So for example, the way I like to do this is that I like to kind of mark my boxes. So if I want to look at yellow smooth, I'm going to put a box around every yellow smooth. Yellow smooth, yellow smooth. And for yellow smooth, for example, we're looking for one big S and one big Y. That's all we need because those are both dominant traits. Oops, I think I did it. What happened? Sorry. Yellow smooth, yellow smooth, yellow smooth. Yellow smooth, yellow smooth. This should be a big S, so that's typo by the book. I like when that happens. So we have, let's count those up. 1, 2, 3, 4, 5, 6, 7, 8, 9. So we have 9 yellow smooth. Then I like to do the other one some other way. So now we're going to look at smooth green. So we have 1, 2, 3 yellow green. Then we have yellow wrinkled. So we have 1 yellow wrinkled. And let's see here. And this is another typo. Oops. Smooth yellow. This should also be little, right? So that's going to be yellow smooth as well. And this should also be yellow smooth. This is not a great figure because there's some mess ups. 3. And then we have our green wrinkled, right? So this is a ratio that you get is a 9 to 3 to 3 to 1 ratio. This is a ratio you must know, just like you need to know 3 to 1 ratio when you're doing 2 hets for a monohybrid. If we're doing 2 hets for a dihybrid, it's going to be a 9 to 3 to 3 to 1 ratio, all right? You have to know that ratio. You have to be able to look at a dihybrid cross and figure that out. You have to be able to draw your own dihybrid cross. All right. So in conclusion, Mendel had these 2 big ideas, all right? He didn't know about genes at the time, but there were genetic factors, we now know they're genes, that can be hidden, right? Especially in those in those heterozygotes that can only express one trait, right? So those hidden traits are going to be the ones that are recessive, right? Despite identical appearance, some dominant can have different genotypes, right? So for example, you can be big P, or big P, little p, and still be purple, right? The trait that is expressed in those F1 plants that is hidden, the hidden recessive trait can reappear when you mate heterozygotes, right? So the 2 big ideas, heredity or inheritance is determined by discrete physical elements for each trait. We now know that those are genes or sequences on a chromosome that can that encode a protein. The second big idea is that diploid genes get separated when you go through meiosis, right? And then regrouped when you come to fertilization, okay? So he had 2 big laws. The first, or it doesn't matter the order, but one we've already talked about, which is this law of independent assortment, where alleles remember, independent assortment is where that occurs in metaphase 1, where you can get homologous chromosomes lining up in any way, right? And then the second one is law of segregation, where alleles get separated during meiosis. This is really going from diploid to haploid, right? And that happens kind of really in anaphase 1 of meiosis 1, right? So those are Mendel's 2 laws. The law of segregation, kind of describing meiosis, going from diploid to haploid in meiosis to make your gametes, and the law of independent assortment, where alleles of the genes can assort independently during meiosis. Okay, so I just want to give a few examples of human traits where we have Mendelian or complete dominance. So an example of that is albinism. So here is a woman with albinism that has very little pigmentation, right? White hair, usually red eyes because of a lack of pigmentation. So inheritance in certain human traits is completely predictable by Mendelian inheritance patterns, right? Exactly like Mendel thought or described. So here, what we have is that we are going to have two heterozygous individuals. These are sometimes called a carrier because they are carrying the recessive allele, but it's usually hidden, right? Because it's recessive. So what are the gametes produced by each parent? So you can either make a big A or a little a. So those are the two possible gametes for the parents. And again, when we make our Punnett square, those gametes go on the axes, right? So for the female and the male. And then what percent of the, and this is a tricky question, what percent of the normal offspring are going to be carriers, right? So we have to look first at only the normal ones, and then we need to see who will be carriers. So that's going to be actually 2/3 are going to be carriers. I always have one question on the test about this, and people always get it wrong. They're like, oh, 25%, right? Or 1/3. And no, I'm not asking about those guys because they're not the normal ones. We're looking at the carriers, right? So if I asked a question, for example, let's do a different question. So what percent will have albinism? That would be 25%, right? Our little 1/3 there. Sorry, 1/4. Sorry. Okay. And again, you can look at genotype and phenotypic ratios. Okay, so let's do an example of a dihybrid cross for human traits. So here we have a gene for pigmentation where you either have normal pigmentation or you can be albino if you're recessive, and you have a gene for hearing. You have big D is normal hearing, little d is deafness. So what is the probability that a couple heterozygous for both genes will have an albino deaf child, right? So before I do this, I always write out my chart. So albino and deaf, right? So I always write out my chart. So there's only 3 possibilities here, right? These two would both be normal. This would be the albino. For the deaf, these both would be normal, and this would be a deaf person, right? And the question asks, what if 2 hets for both genes got together? What are the chances that you would have an albino deaf child? So we are dealing with a cross where we have heterozygous for albino and heterozygous for deaf, cross to heterozygous for albino and heterozygous for deaf, right? First thing first, you always want to figure out what are the gametes. Remember that the gametes are going to have 1 copy of each gene, or each allele rather, right? So again, it's going to be a 4x4 Punnett square, right? But hold on, do we have to draw that whole thing out? No, because we know what the predicted ratio is. And the question asked, okay. What is the probability a couple will have an albino deaf child, right? So these guys would be all normal, right? These ones would be albino, okay hearing. These ones would be deaf and normal pigment. And here we would have deaf, albino. All right. So we know without even, oh my goodness. Whoa, what happened there? Hold on, let me go back. Open the wrong app. All right, so we don't even have to do the cross if we know this ratio when we have two doubly heterozygous mated together, right? So the chances would be 1 in 16 for our deaf albino person. I want to show you one other cool way to do this, right? And I don't care how you do it, as long as you show your work. So another way to do this, so we had albino and deaf, right? Yep, okay. So here is our cross. Another tricky way to do this is to break them up and look at one gene at a time. So if we only look at the A's, we would have a Punnett square that looked like this, right? And what is the chance of having an albino child here? Would be 1/4. And then we can do the separate for deaf. And what are the chances of having a deaf child here? 1/4. We're going to multiply those frequencies together to give us our frequency of having an albino deaf child, right? What if we wanted to ask probability of having a normal pigmented deaf child, right? And if we did that, we would have, so the normal pigmented would be 3/4 times 1/4. We would get 3/16, right? All right. And so we are going to leave it at that. And we pick up kind of pedigrees in the next ones. Okay. Peace out. Oh, where's my, there we go.